All right, welcome to The Explainer. Today, we are going deep inside the microscopic city that is a single cell, and we're going to track one of its most essential workers, a protein. I mean, think about that question. Every one of your cells runs a logistics network, more precise, more complex than anything we've ever built on Earth. So how does it do it? Well, to figure that out, we're going to follow the epic step-by-step journey of just one single protein, from the moment it's created to its final crucial destination. Okay, so our story kicks off right here, in the cell's massive, bustling manufacturing plant. It's this huge network of membranes called the endoplasmic reticulum, or as we'll call it, the ER. So here's the deal. All proteins, every single one, start being built out in the cytosol. That's kind of the jelly-like goo filling the cell. But, and this is key, only certain proteins are meant for this special pathway. The secret is a unique address label, a little tag of amino acids called a signal peptide. This incredible idea, the signal hypothesis, is what explains how the cell knows which proteins get a VIP pass into the ER. And the organization here, it's just mind-blowing. Look at this protocol. As soon as that signal peptide pops out of the ribosome, this molecular bodyguard, the signal recognition particle, or SRP, grabs it, and boom, production halts. The SRP then escorts the whole thing to a specific docking port on the ER, plugs it into a channel called the translocon, which is basically a secure gateway. Only then does the factory start back up, feeding the protein directly inside. It's flawless. Now, once our protein is safely inside the ER's inner space, the lumen, the work is far from over. First, that signal peptide gets snipped right off. Its job is done. But now you've got this long, floppy chain that has to fold into a perfect 3D shape, and you better believe this isn't left to chance. These little helpers called chaperone proteins, like one named BIP, come in and guide the chain, making sure every fold is perfect, because without that exact shape, our protein is totally useless. All right, our protein is folded and ready to go. So what's next? Well, it's time to move from the factory floor to the finishing and packaging department, this amazing stack of flattened sacks called the Golgi apparatus. You know, this thing was first spotted by its namesake, Camillo Golgi, way back in 1898. He just called it an internal reticular apparatus. Today, we know it's basically the cell's central post office. It has a receiving dock, the sysface, where packages from the ER arrive, and a shipping department, the transface, where the finished products get sent out. Inside the Golgi, our protein gets this incredible, super precise chemical tune-up. Seriously, think of it like a high-tech pit crew for a race car. As the protein moves from one Golgi stack to the next, different enzymes go to work in a very specific order. They'll snip off some sugar molecules, biologists call them residues, and add on others. This whole customization process, called glycosylation, is absolutely critical for what the protein will do and where it's going to end up. Now, the way the Golgi sorts everything, it's just ingenious. In our case, this specific protein is heading to the lysosome. So the Golgi slaps a very particular tag on it, a phosphorylated mannose sugar. This little tag, mannose 6-phosphate, is basically a high-priority, destination-specific shipping label. It guarantees our protein gets put in the right box for delivery. So, our protein is folded, it's been customized, and now it's been tagged. It's ready for shipment. But how does the cell actually, deliver the packages? It uses these amazing little delivery trucks made of membrane called vesicles. And the whole process is this beautiful dance of what we call budding and fusion. A vesicle literally buds off from the Golgi membrane, wrapping our protein up inside. It then travels through the cell and fuses with its target. The membranes merge, and the contents get delivered. Simple, elegant, and incredibly effective. And this isn't just happening by accident. It is a highly regulated 4-step process. First, the cargo gets selected based on those tags, like our mannose 6-phosphate. Second, special coat proteins gather around and help pinch the membrane off into a perfect little bubble. Third, that vesicle zips along the cell's internal highway system, the cytoskeleton. And finally, specific proteins called snares act like a molecular lock and key, making sure the vesicle docks and fuses only with its one correct destination. The Golgi can also ship things right out of the cell completely, and it does this in 2 main ways. You've got your steady, continuous stream of deliveries. That's constitutive secretion. But then, for important stuff like hormones, the cell uses regulated secretion. It stockpiles the protein in vesicles and only releases them when it gets a specific go signal. It's an on-demand delivery service right inside you. And now, after all that, thanks to its special tag, our protein has been sorted, packaged, and shipped. It finally arrives at its destination, the lysosome, the cell's powerful demolition and recycling plant. The first thing you need to know about this place is that it is super acidic inside, with a pH of about 4.5. And that acidic environment is everything. Why? Because the lysosome is packed with dozens of powerful digestive enzymes called acid hydrolases that only switch on and get to work in a highly acidic bath. What's really wild is that lysosomes are polymorphic. They actually change their form. When it's first made, it's called a primary lysosome, and it's basically just a storage bag full of sleeping, inactive enzymes. But once it fuses with a vesicle carrying junk to be digested, it transforms into a secondary lysosome, a churning, active digestive powerhouse. And don't think for a second this is just about garbage collection. No, no, no. These demolition crews are vital for recycling the cell's own old and worn-out parts, a process called autophagy. They're involved in the programmed destruction of the cell itself, and, amazingly, they even play a huge role in fertilization when a sperm cell releases its lysosomal enzymes to burrow into the egg. With all those corrosive enzymes inside, containment is absolutely everything. That single membrane wall has to be incredibly tough. Things like cholesterol actually act as stabilizers, reinforcing the membrane and making it stronger. But other things, called labelizers, can weaken it. A leak? Oh, that would be catastrophic. The cell would literally start digesting itself from the inside out. And if you ever doubted how critical this whole pathway is, just look at what happens when it breaks. If just one of those dozens of lysosomal enzymes is missing because of a genetic defect, the waste it's supposed to break down just builds up. It fills the lysosome, causing a whole group of devastating illnesses we call lysosomal storage diseases. And so, the journey is complete. From a simple blueprint in our DNA, our protein was built in the ER, customized in the Golgi, and then delivered with absolute pinpoint precision to the lysosome to do its job. It's a perfect illustration of the dynamic, highly organized city that's at work inside every single one of our cells. The sheer elegance of this system is just astounding. The signal peptides, the molecular tags, the vesicle delivery trucks. It makes you wonder, right? If all this complexity goes into the journey of just one protein, what other incredible secrets are our cells still waiting for us to uncover?